Transcription is the process of producing a strand of RNA from DNA using DNA as a template. DNA molecules contain 4 different nitrogenous bases arranged in specific sequences that provide the information for producing specific proteins. During transcription, a sequence of bases from DNA is transcribed onto a strand of RNA, passing genetic information from DNA to RNA. The RNA produced is messenger RNA, mRNA, which carries instructions for protein production. A gene is a specific segment of DNA whose base sequence determines the sequence of nucleotides in the RNA molecule. For each gene, only the DNA strand, called the template strand, is transcribed, while the other strand can serve as a template for different genes. RNA polymerase is the enzyme responsible for separating the 2 DNA strands and synthesizing mRNA. It adds complementary nucleotides, adenine, uracil, cytosine, and guanine, to the DNA template strand. Transcription begins when the RNA polymerase binds to the promoter region of the DNA, initiating synthesis of mRNA along the transcription unit, where RNA polymerase attaches, unwinds the DNA, and synthesizes mRNA as the DNA passes through the enzyme. As mRNA are formed, it detaches and the DNA strands rejoin to reform the double helix. In prokaryotes, transcription ends when RNA polymerase reaches a terminated sequence, causing the enzyme to detach and release the mRNA transcript. No further processing is required, and translation can begin immediately, even while transcription is still in progress. Translation is the process where the nucleotide sequence of mRNA is used to build a protein. The mRNA code is read in groups of 3 bases called codons, each specifying 1 amino acid. With 4 RNA bases and 64 possible codons, these combinations code for 20 amino acids. Translation occurs at the ribosomes, which have 3 sites: A, aminoacyl, P, peptidyl, and E, exit. tRNA molecules, each carrying a specific amino acid, bind to the complementary RNA codons using a 3-base anticodon. The ribosome moves along the mRNA, and the amino acids are joined together to form a polypeptide chain, which folds into a functional protein. Translation begins with a start codon and an initiator tRNA carrying methionine. It ends when the stop codon reaches the ribosome. A release factor binds, releasing the completed polypeptide. Multiple ribosomes can translate the same mRNA sequence simultaneously, increasing protein production. After translation, proteins often fold and undergo modifications. Example, addition of sugars or lipids. Messenger RNA, mRNA, and transfer RNA, tRNA, are essential intermediaries in protein production, converting genetic instructions from DNA into polypeptides so cells can produce proteins for growth, function, and adaptation. mRNA carries genetic information from DNA to the ribosome. It contains a 5' cap for stability and ribosome binding, a codon region, protein blueprint, a 3' UTR, regulatory role, and a poly-A tail for stability and export. Before functioning, mRNA undergoes processing, capping, protection from degradation, splicing, removal of introns, and polyadenylation, addition of the poly-A tail, producing a mature transcript. tRNA has a cloverleaf shape with an anticodon region that pairs with mRNA codons and an acceptor stem that binds a specific amino acid. Its D-arm supports structural stability, and its T-arm enables ribosome recognition. Acting as an adapter, tRNA translates the genetic code by delivering the correct amino acids to the ribosome for accurate protein assembly. During translation, ribosomes move along the mRNA, matching codons with tRNA anticodons to build the polypeptide chain efficiently and accurately. Cells maintain quality control by degrading faulty mRNAs, checking amino acids before attachment to tRNA, and monitoring codon-anticodon pairing. Gene expression is further regulated at the transitional level by microRNAs, RNA binding proteins, and enzymes that modify mRNA stability or tRNA function to ensure proteins are produced only when needed. Without mRNA to transfer genetic instructions and tRNA to deliver the correct amino acids, the genetic information stored in DNA could not be translated into functional proteins. Polypeptide synthesis is important for many reasons. It creates essential proteins such as enzymes, hormones, antibodies, and structural proteins. Proteins regulate nearly every cellular function and therefore are essential for life. Polypeptide synthesis allows for genetic expression and variation where only specific genes are activated to meet the cell's current needs. Also, variation allows for evolution and adaptation. Accurate polypeptide synthesis ensures proteins fold correctly and function properly. Errors in protein production or gene expression can contribute to diseases such as cancer. Polypeptide synthesis is fundamental in all life because proteins act as enzymes that catalyze metabolic reactions, structural components that maintain cell integrity, signaling molecules that regulate physiological processes, and immune molecules such as antibodies that defend against pathogens. It is responsible for overall growth, repair, maintenance of cells, and phenotypic expression. The majority of complex traits are controlled by many genes, each contributing a small amount to the final phenotype. The genotype is the fundamental instruction manual for an organism. External factors can switch genes on or off, or alter the proteins that they produce. Physical factors such as light and temperature can drastically change appearance. An example is Himalayan rabbits that develop dark fur only in cool places of their body. Genes encoding enzymes involved in melanin production determine pigmentation. Environmental factors such as temperature can alter enzyme activity, influencing the phenotype expressed. Chemical and nutritional factors such as diet can determine the life path of an individual. For example, honeybees that are fed royal jelly as larvae become queen bees, whereas bees on a normal diet are worker bees. Social and behavioral factors can also affect phenotypic expression. For example, in some reptiles, the sex of the offspring is determined by the incubation temperature of the eggs rather than the chromosomes. Therefore, polypeptide synthesis is critical because it converts genetic information stored in DNA into functional proteins that can determine cellular structure, metabolic activity, and ultimately the phenotype of the organism.